Okay, we're going to continue on with Chapter 4, the cell. All organisms are made of cells. The cell is the simplest collection of matter that can be alive. All cells come from other cells. Those cells can differ substantially from one another. They share many common features. Most cells are between 1 and 100 micrometers in diameter, which is too small to be seen by the unaided eye. So we use microscopes to view cells. We didn't talk too much about the microscopes, but we did, however, use them. So we're going to skip over all the microscope stuff, and we're going to start with eukaryotic and prokaryotic cells. So the basic structural and functional unit of every organism is one of two types of cells. They're either prokaryotic or they are eukaryotic. Organisms of the domain, bacteria and archaea, consist of prokaryotic cells. So all bacteria are prokaryotic, all prokaryotes are bacteria. And then that leaves your eukaryotes, your protists, your fungi, your animals, your plants. All cells share some basic features, including the plasma or cell membrane, semi-fluid substance called cytosol, chromosomes which carry your genes, and ribosomes which make proteins. Prokaryotic cells are characterized by having no nucleus. Their DNA is in an unbound region called the nucleoid. They have no membrane-bound organelles, so they don't have any ER, they don't have Golgi, they don't have mitochondria, they don't have chloroplasts, they don't have lysosomes, just to name a few. They do have cytoplasm, which is bound by the plasma membrane. So here is your typical prokaryotic cell. Now this prokaryotic cell has DNA in a nucleoid region. It has fimbriae, which allows it to adhere to substances. The outermost part is the capsule. Not all prokaryotes have capsules. So the capsule just provides an extra layer of protection for the bacterial cell. Within that capsule there is a cell wall, and within that cell wall is a cell membrane or plasma membrane. Notice that the bacterial cell also has multiple flagella which allow it to move. Eukaryotic cells are characterized by having DNA in a nucleus that is bounded by a membranous nuclear envelope, and membrane-bound organelles, many of the ones that I just mentioned, your mitochondria, Golgi, ER, lysosome, chloroplasts. They also have cytoplasm in the region between the plasma membrane and the nucleus. Eukaryotic cells are generally much larger than prokaryotic cells. The plasma membrane is a selective barrier that allows sufficient passage of oxygen, nutrients, and waste to service the volume of every cell. The general structure of a biological membrane is a double layer of phospholipids. So here is the plasma membrane. It is a double layer of phospholipids. There are phospholipid tails, which are hydrophobic, which point toward each other. And there are hydrophilic heads, which point inside and outside of the cell. Okay, this looks like it's outside of the cell, and this is inside. Now, embedded within that bilayer is proteins. Okay, here you have a channel protein, here you have some other proteins. These proteins have many diverse functions, which we will get to in Chapter 5. Notice that some of the proteins also have carbohydrates attached to them, which leads to a function of cell-to-cell recognition. Every different type of cell will have different carbohydrates or glycolipids and glycoproteins on the surface of their cells. So the portion of the cell membrane that points out and in is hydrophilic, because that's the portion of the membrane that is in contact with water. Metabolic requirements set upper limits on the size of cells. The ratio of surface area to volume of a cell is critical. As the surface area increases by a factor of n squared, the volume increases by a factor of n cubed. As a result, small cells have a greater surface area relative to their volume. This is going to be very important because the cells must get enough stuff, material, molecules into the cell, ions into the cell, and it must also be able to release waste from inside the cell to outside the cell. So the cell has to have a large surface area to volume in order to be able to facilitate the movement of all these molecules. So this is showing an example of what happens to the surface area to volume ratio as the cell or this box increases in size. So here, total surface area of this small box is 6, and the total volume, height times width times length, is just 1. So the surface area, 6 to volume, 1. Okay, so surface area to volume ratio is 6 to 1. But if the... sorry, my child is yelling for me. As the box or the cell increases, what happens is the volume increases at a higher rate than the surface area does. So that leads to a much smaller surface to volume ratio. That's bad for the cell. The cell can't get stuff in and get stuff out to satisfy all the metabolic requirements of the cell. So what would have to happen is that cell would need to divide into many smaller cells. A eukaryotic cell has internal membranes that divide the cell into compartments called organelles. Each of these organelles has a specific function. The plasma membrane and organelle membranes participate directly in the cell's metabolism. So this is a eukaryotic cell. This is an animal cell. So notice the very large visible nucleus. Outside of that, you have ER, you have rough ER, and you have smooth ER. You have the mitochondria. You have, let's see what else, you have lysosomes, you have the Golgi, you have ribosomes, which are both free in the cytoplasm and bound to the ER. And then, of course, you have your cytoskeleton, which functions to, for example, allow organelles to move along it. And then here's your plant cell. Notice that your plant cell has a very specific structure due to the cell wall, the rigidity of the cell wall. In addition to that, you see a large central vacuole here. You see plasmodesmata, which are basically pores in the cell wall. Notice that you also have chloroplasts, which allow the plant cell to undergo photosynthesis. Okay. The nucleus contains most of the DNA in a eukaryotic cell. Ribosomes use this information from the DNA to make proteins. The nucleus contains most of the cell's genes and is usually the most conspicuous organelle, as you saw in the picture. The nuclear envelope encloses the nucleus, separating it from the cytoplasm. The nuclear membrane is a double membrane. Each membrane consists of a lipid bilayer. There are pores within that nuclear envelope that regulate the entry and exit of molecules from the nucleus. The shape of the nucleus is maintained by the nuclear lamina, which is composed of protein. So here's your large conspicuous nucleus, which the nuclear envelope is continuous with the rough ER. In the nucleus, DNA is organized into discrete units called chromosomes. Each chromosome is one long DNA molecule associated with proteins. As you know, we have 46 chromosomes in our body or somatic cells, and we have 23 chromosomes in our gametes, egg and sperm. The DNA and proteins of chromosomes are together called chromatin. So remember that the DNA wraps around histones. Those are the proteins. That's a specific name for the proteins that the DNA wrap around. Chromatin condenses to form discrete chromosomes as a cell prepares to divide. The nucleolus is located within the nucleus and is the site of ribosomal or RNA synthesis. Ribosomes are complexes of ribosomal RNA and protein. Ribosomes carry out protein synthesis in two locations. Free ribosomes are found in the cytosol, and bound ribosomes are found attached to the endoplasmic reticulum. So free ribosomes are going to make proteins that will stay within the cell, have specific functions in the cell, and your bound ribosomes will make proteins that will be exported out of the cell. Okay, so here's your microscopic image of the rough ER and ribosomes. The endomembrane system consists of the nuclear envelope, the ER, the Golgi, lysosomes, vacuoles, and the plasma membrane. They are all connected by either being continuous with each other or through transfer by vesicles, the little vehicles that carry things throughout the cell. The ER accounts for more than half of the total membrane in many eukaryotic cells. The ER membrane is continuous with the nuclear envelope. There are two distinct regions, the smooth ER, which doesn't have ribosomes, and the rough ER, which has bound ribosomes. Here's another picture of that. Your nuclear envelope, continuous with your ER. Here is your rough ER, which has ribosomes. Here's your smooth ER, which does not. The two have very different functions. Functions of the smooth ER include synthesis of lipids, metabolism of carbohydrates, detoxification of drugs and poisons, and storage of calcium ions. The rough ER has bound ribosomes, which secrete glycoproteins, proteins covalently bonded to carbohydrates. They distribute transport vesicles, proteins surrounded by membranes, and the rough ER is also a membrane factory for the cell, makes membrane. The Golgi apparatus consists of flattened membranous sacs called cisternae. Functions of the Golgi include modifying products of the ER, manufacturing certain macromolecules, and sorting and packaging materials into transport vesicles, which will be destined for export out of the cell. So here's your Golgi. There's the cis face, which is the receiving end that receives products of the ER, and then there's the trans face, which is the shipping end, which will ship products off to the cell membrane. A lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. Lysosomal enzymes can hydrolyze proteins, fats, polysaccharides, and nucleic acids. Lysosomal enzymes work best in the acidic environment inside the lysosome. Some types of cell can engulf another cell by phagocytosis, which forms a food vacuole. The food vacuole will then fuse with the lysosome, and the lysosome will release its digestive enzymes, and the macromolecules will be digested. So here's an example of phagocytosis, where you have food being ingested by a phagocytic cell, and that food forms a food vacuole. The ingestion of that food forms a food vacuole, which then fuses with a lysosome, which releases its digestive enzymes to break down those molecules, and that is digestion. Here's autophagy, where basically your lysosomes are destroying like old organelles or organelles that don't really work that well anymore. Vacuoles are large vesicles derived from the endoplasmic reticulum in the Golgi apparatus. Food vacuoles are formed by phagocytosis. Contractile vacuoles are found in many freshwater protists, and they pump excess water out of the cells. So remember that when the freshwater protists are in fresh water, their environment is hypotonic. And inside the protist is hypertonic. So what happens is water rushes into the cell because there's more solute found on the inside of the protist than in the surrounding fresh water. So in order to adapt to that problem, what they do is they have this contractile vacuole which pumps excess water back out, maintaining water and solute balance between their cell and the environment. Central vacuoles are found in many mature plants, and they hold organic compounds in water. Certain vacuoles in plants and fungi carry out enzymatic hydrolysis, just like lysosomes. And here's your large central vacuole. This just, once again, goes over your endomembrane system, the parts, and how they're connected. So here we have DNA found within the nucleus, which contains instructions for making protein, which may be made by ribosomes found on the rough ER, which then get transported to the Golgi, which then get packaged and modified, and then sent to the plasma membrane for export outside of the cell. Okay, that'll conclude part 1.